Hello students, welcome to George Biology. Number B, plastids. If we talk about plastids, they are observed in plants and in some members of Protista like euglenoids. So plastids, they are unique to plants and they are also observed in euglenoids. Now this different type of plastids, their nomenclature that was given by Schimper. Now different type of plastids, they are originated from a basic material which is called as proplastid. So here the initial structure from which all the plastids are developed, that is proplastid. Now this proplastid is a primitive one from which different type of plastids have been originated. The first plastid which is developed from proplastid, that is chloroplast. And this chloroplast that consists of green pigment and that is called as chlorophyll. So it consists of a predominant pigment, that is chlorophyll. Along with that, it also consists of carotenoids also. Now this one, it helps in photosynthesis. And due to presence of chlorophyll in it, it is green in color. Most of the mesophyll cells and the green parts of the plants that consist of chlorophyll in it. Now apart from this chloroplast, it also helps in formation of certain colorless plastids, and that is leukoplast. And this leukoplast, it is a colorless plastid. It's a colorless plastid. It means it does not consist any pigment, so no pigments are present. It does not consist grana, so it is agranal. Then what is its function? It helps in storage. That is why it is also called as storage plastid. Now what does it stores? Based on the type of material which it stores, these are of different types further again. So here, if it helps in storage of oils and fats, especially in the oily seeds, these are present, then such kind of plastids, they are called as eleoplast. And the good example for this one, that is endosperm of castor, you can take. Next one, if it helps in storage of proteins, now that is called as proteinoplast or else ailuroplast. Ailuroplast. And the good example, that is aileron layer of maize plant. And the last one, if it helps in storage of carbohydrates or else starch, now that is called as amyloplast. Amyloplast. And the good example, that is potato tubers you can take, which is starch rich, or else rice grains you can take. Now this is a very important one. Based on the utilization of this simple framework, we can make multiple types of questions. For example, out of the following, which is odd with respect to the storage plastids? Eleoplast, ailuroplast, amyloplast, chromoplast. You can take as chromoplast. Now apart from these two different type of plastids, there is one more type of plastid that is present, which is formed from the maturation of this chloroplast and leukoplast. So here, if this chloroplast and leukoplast that gets matured, now that helps in formation of one more type of plastid, that is chromoplast. In the name itself, you can find the explanation, chroma, right? That means color. So these are colored plastids. They may consist of yellow color, orange color, or sometimes red color also. And this coloration is due to presence of fat soluble substances or pigments that is called as carotenoids. And these carotenoids, it's the name of the group which consists of two different pigments, that is carotene and number 2 xanthophyll. Apart from all these things, there's a small special property is also there. Sometimes this proplastid, by production of certain pigment called as etiolin, it becomes into etioplast. Etioplast. Etioplast, it consists of etiolin pigment. Etiolin pigment. Now due to presence of this etiolin pigment, it helps in internodal elongation. But sooner or later, it converts again into chloroplast. Now these are the different types of plastids. Let's go with the most important aspect from the plastid topic, that is chloroplast. Now if we talk about chloroplast, we already said it's a photosynthetic one. It helps in photosynthesis due to presence of a pigment called as chlorophyll in it. Now this chloroplast, it can be varied in number in different cells. For example, it can be 1 per cell in case of Chlamydomonas, or else it can be 20 to 40 per cell in case of green algal members. Now they are varied in their shape as well. They can be lens shaped, oval, spherical, discoid, or else sometimes ribbon like structures as well. And this is also a double membrane bound cell organelle like mitochondria. As we said that it consists of double membrane bound structure. Let's take the picture here. So this is the outer membrane, and this is the inner membrane. Let's start with the outer membrane. So here is the outer membrane. Now this outer membrane, it is rich in porins. It is rich in porins. It consists of phospholipids, and it is semi-permeable in nature. It is semi-permeable in nature. Number 2, inner membrane. Now this inner membrane, it consists of sulfolipids, and it also consists of galactolipids. Sulfolipids as well as galactolipids that are present. Now due to presence of these two materials, it makes the membrane almost impermeable. So if a question is asked with respect to the permeability in mitochondria as well as chloroplast, which is more permeable, which is less permeable? Outer membrane is more permeable, inner ones is less permeable. In both the cases, the property is same. Now, so two membranes we have taken. Apart from these two membranes, there is a third membrane system is also present in the inside, and that one is called as thylakoid. So here the unit, the single unit is called as thylakoid. And this thylakoid, it consists of a space inside which is called as lumen. And these thylakoids, many of them, they pile up and helps in formation of a bundle. And that bundle of thylakoid is called as grana. So what is grana? It's a bundle. It's a bundle of thylakoid. Right? That is called grana. So this entire ones. Now such kind of grana, many are present here, and these many grana, they are connected by one more tubular structure, and that tubular structure, it is called as stroma laminate, or else is also called as stroma frets. Apart from this one, there is a liquid material is also present, and that liquid material is called as matrix or else stroma. So what are the things which are present in the stroma region? It consists of double-stranded circular DNA. It consists of double-stranded circular DNA. Number 2, it consists of 70S ribosomes. It means here also, in case of chloroplast, there will be synthesis of proteins takes place. And number 3, it also consists of certain enzymes which helps in photosynthesis, and these enzymes, they are called as Rubisco. So it consists of certain enzymes which helps in photosynthesis, that is Rubisco. Ribulose 1,5-bisphosphate carboxylase and oxygenase. Now this Rubisco, it's a dual enzyme. It's a dual enzyme, and it's a most abundant enzyme on the earth. What is this dual activity of Rubisco? It consists of carboxylase activity as well as oxygenase activity as well. Let's see, right? So what is this activity that it consists? It consists of carboxylase activity. Carboxylase activity. When this carboxylase activity will be triggered? When there is high CO2 concentration is present in the atmosphere and low O2 concentration. So at high CO2 concentration, it helps in fixing CO2. When it fixes the CO2, it helps in formation of glucose, will form glucose, and this process we call it as photosynthesis. Photosynthesis. On the other hand, if there is less amount of CO2 is present and more amount of O2 is present, now at that time it helps in fixation. It helps in fixation of O2 and it removes CO2. Now if there is a removal of CO2 if takes place, now that leads to decrease in glucose production. And this property is called as photorespiration. Photorespiration. When this happens, whenever the enzyme triggers the oxygenase activity. Functions. As we have already discussed, it helps in photosynthesis. It helps in photosynthesis. Slight connection will take, which can help us in photosynthesis chapter also. So this photosynthesis, it takes place in two different steps. Number one, that is light reaction, and number two, dark reaction. Now where does this light reaction takes place? Now this light reaction, it takes place that is in Grana and stroma frets. Stroma frets. It means here itself we are clearing what is the site of light reaction, that is Grana and stroma frets or stroma laminae. Number two, then what about dark reaction? It takes place in stroma region. Now apart from this function, it also helps in storage of starch. Now this is all about the semi-autonomous cell organelles.